Hello everybody, it's Miss Greaves. Today we're going to be looking at chemical agents of control, getting ready for your laboratory this week. And before we get started, I'll show you this little video clip to get you rocking and rolling. So what was the little Muppet at the end there doing? Any guesses? Yeah, he was spraying cleaner. He was spraying a disinfectant on the toilet. They happen to be pretty big reservoirs for bacterial growth, and bacteria can replicate quite rapidly, 20 minutes. Sometimes they can replicate as fast as 20 minutes. So within 12 hours, you got a population of 1 billion different bacteria. So what are the ways that we can control these? And this is what we're going to be looking at in this week's lab, and it's going to be very relevant to your creating your emergency plans. We're going to be looking at the efficiency of antiseptics and disinfectants on two different bacterial populations. So before we get started, it's important to note that there are slight differences between antiseptics and disinfectants. Most people use the terms interchangeably, but they are in fact slightly different. Antiseptics are chemicals used to kill or inhibit the growth of microorganisms on tissue. So those would be things like mouthwash or maybe diluted ethyl alcohol that you might clean a scrape or a cut with. They're not very strong. They're not going to break down your tissue. Disinfectants are actually quite powerful, and they destroy microorganisms in water, in air, or on surfaces. So they tend to be a little bit stronger because of that. So your water supply, when it comes from the water treatment facility, has actually been treated with chlorine. And they do this to make sure that there's no living pathogens that have passed through any of the other filter systems. So in our lab tomorrow, we're going to be looking at the factors that influence disinfectant effectiveness. And there are several factors that you want to consider when you're looking at disinfectant effectiveness. So what type of microbial population are you going to be destroying or trying to destroy? So we're going to be looking at a couple different populations. So we're going to be looking at Escherichia coli, more commonly known as E. coli, which lives in all animals, the guts of all animals. And it's actually quite handy because it helps you break down undigested particles of food and uptake those into your intestinal system. We're also going to be looking at Staphylococcus aureus, so staph, we can just call it staph. And one of the things you'll notice about this picture is that the staph is actually colored a purple color, and that's because the Escherichia, the E. coli, actually has slightly different cell membrane structures. And it's important to note that the type of pathogen or microorganism that you're trying to destroy, they're going to have different types of evolutionary adaptations, and you have to take those into consideration when you're trying to control them. So a lot of bacteria that are what's called gram-negative, like E. coli, they actually have something in their cell wall that helps, in their cell membranes, that actually helps them to repel the effectiveness of antibiotics. So that's where you get some of those antibiotic-resistant strains of bacteria. They have different adaptations or different traits. So the concentration of your disinfectant can also be a factor of how effective it is. If you put 1 drop of chlorine in 5,000 gallons of water, that's probably not going to cut it. So you have to consider concentration. The length of exposure, so that the amount of time that a pathogen is exposed to a disinfectant can have an impact on its effectiveness also. There's also other conditions and environmental conditions that we won't be looking at tomorrow. So the pH of the environment, or even the temperature. If the temperature is slightly elevated, the disinfectant might actually be more effective. And if there's a potentially or a slightly higher pH, that can sometimes actually break down the disinfectant and actually make it less effective. So these are just types of things you want to consider when you're looking at how you're going to use chemical agents of control in your emergency plan. Oops, let's move on. So let's take a look at some ways that we can develop your controlled experiment. Now, we've done controlled experiments before, and you should have a pretty good idea that the reason you do a controlled experiment is you're looking for cause and effect. You're looking for you manipulate one variable, you get an impact or an effect on another variable. So you have dependent and independent variables. The dependent variable is going to be the one that you're placing the treatment on or changing. So take a second to think about, if you're in our controlled experiments tomorrow, where we're going to be looking at the impact or the effectiveness of disinfectants on a population of bacteria, which one's the dependent variable and which one would be the independent variable? Well, hopefully you got that right. If you said that the independent variable was the disinfectant or the time, and that the dependent variable was the bacterial population, then you'd be correct. So if you didn't get that right, you might want to check out or review our video on our class website titled True Experimental Design. Okay, that's going to kind of review some of those key concepts. You also want to have a control in your experiment. Why do we want a control? Well, let's say that you picked up your bacterial culture from me, and you ran your experiment, you had a control you had a control quadrant, and you had all your treatments. And then 2 days later, when you went to look at your petri dish, there was absolutely no bacterial growth anywhere, not on those treated with disinfectants, and it was not in the control. So that means that bacterial growth wasn't necessarily the fact that there was no growth wasn't due to the disinfectant. It was due to maybe the fact that your bacterial culture was incorrectly completed. Okay, so you need to have a control to make sure that the changes you see are in fact caused by the variable that you're changing. Okay? So when you come into the lab tomorrow, you should have a pretty good idea of what you might want to study, or whether you want to study the different types of disinfectants, or whether you want to look at the effect of exposure, length of time. Okay? You must work with your team members, and everyone needs to participate and come up with a very strong experimental design with procedures. You must bring it to me for approval. Okay, if you're having problems, that's why I'm here. Don't worry, if you still are a little fuzzy on this, we'll talk about it and brainstorm when we get into class tomorrow. We still should have a pretty good idea of controlled variables, independent and dependent variables. Once you get your design approved, you're going to gather your supplies: gloves, goggles, typical lab safety material, sterile swabs, 2 to 6 sterile test tubes, pipettes, and disinfectants, and glassware marking pens. Now, once you have all your supplies, come to me, and I will be giving each member of your team 2 petri dishes full of nutrient agar. You've worked with this before. It's where the bacterial cultures are swiped or streaked, and that's where the bacteria will grow. Okay, you're going to want to label the petri dishes. Again, it's going to maybe change, maybe vary slightly depending upon your experimental design, but you'll have a control area. You'll have 3 treatment areas, and each petri dish is going to be testing on different bacterial population. Okay, either E. coli or the Staphylococcus. Okay, you got to put your name on there as well. So this is how I suggest that you label it. When you get your petri dish, you're going to flip it upside down. So the nutrient agar will be stuck on the bottom. So you're going to flip it upside down, and you actually label directly on the bottom of the petri dish. The reason we do this is because if you label the top, you would not be able to see bacterial growth. Once you conduct your experiment and streak the bacterial cells, you do not want to open up your petri dish anymore. You don't want to introduce either bad bacteria out of the petri dish or any bacteria into the petri dish. So you can see on here how it's divided into 4 sections. You've got your control, and you've got treatment 1, treatment 2, and treatment 3. And then the name of the bacteria is also on the bottom of the petri dish. Make sure you put your name on the side of the lid, okay? So remember, we've worked with bacterial cultures before. Once you have your petri dish properly labeled, I will give you your bacterial cultures, the E. coli and the Staphylococcus. Each time you use you're placing a bacterial culture on your petri dish, you need to remember to use a sterile swab. So you're only using the swab 1 time. You don't want to introduce any other contaminants into your petri dish, okay? Once you streak your control, see in this zigzag formation that I have in this picture here, then you're going to add your treatments. So you're going to add 5 milliliters using your pipette of the selected disinfectant, or if you're studying multiple disinfectants, to sterile test tubes. So again, if you're going to study 3 different antiseptics, you need 3 test tubes. Each of those need 5 mils of the disinfectant in them. Then you're going to add 0.5 mils of the culture to the 5 mils of disinfectant. Okay, and you're going to kind of swirl it around, make sure it all gets mixed up pretty uniformly there. You kind of tap the test tube on your hand. Okay. Again, those of you who, if you want to study the effectiveness, or excuse me, the exposure, time exposure, you're not going to need multiple. You're not going to need multiple test tubes. You're only going to need 1 test tube. And when you add the culture to the disinfectant, you will swipe it onto the petri dish at differing time intervals. Okay, so you don't need 3 different test tubes. You only need 1. So it really depends on your experimental design. Okay, I know this might seem a little confusing now, but it will really be cleared up once you have your experimental design locked in. Okay. Now this is important. Once you place all your bacterial cultures and your treatments onto your petri dish, you want to tape it shut and invert it, turn it upside down, and put it in the incubator. And then we'll check them. We're going to be looking at them in about 2 days. You guys can count bacterial growth and see if it's different in the different treatment quadrants. Okay. Before the lab tomorrow, I want to really make sure you guys are prepared, just like any lab. I want you to go to the class website, and I want you to take there's a posted online quiz called Chemical Agents of Control, and it's going to have various questions concerning factors influencing disinfectant effectiveness, and it will also have questions related to experimental design. Okay, so you should be able to answer 80% of those correctly. If you can't, you should go into our class library and take a look at the videos on experimental design and chemical agents of control. Okay, you can and also watch this video one more time. This should be a really fun lab. It's always really neat to see what you guys get on your petri dishes, and then you guys will be able to compare between groups and see which disinfectants are effective, and maybe some of them aren't very effective. Okay, so make sure you can come into class prepared, and hopefully this video has given you some ideas of the types of ways that you might want to set up your experiment. All right, see you guys in lab. Bye.